when you're ready.
Stem cell therapies, are they effective? Despite the fact that stem cell therapies are currently ineffective, this could change in the future as studies show promising results. Why are stem cell therapies important? Bones, organs, and systems all grow and are maintained by the help of human stem cells. If stem cell research is effective, this can help many other researchers around the world who are looking for cures and treatments to certain diseases. There are many issues regarding the use of stem cell therapies, and we will be addressing these through the ethical, scientific, and futuristic lens. We decided not to use the economical lens in our presentation as we thought it did not add to the question, are stem cell therapies effective? Now, Anne-Marie will be talking through the scientific lens.
Before we can start talking about the current science to do with stem cells, we first need to talk about what stem cells are and the ways they're formed. There are two types of stem cells, HESCs, human embryonic stem cells, and HASCs, human adult stem cells, and they are each formed in two separate ways. The first way of formation of HESCs are human embryos that have already been conceived. The second way of formation is somatic cell nuclear transfers, which is a process of cloning. However, it destroys the embryo. The second HASCs are formed through two separate ways. Developed tissues, which only produce a few number of HASCs and only produce that same type of HASC. Take, for example, liver cells. They will only produce liver cells. The second type is induced pluripotent stem cells, iPSCs, which is an experimental modification that is made to an HASC to make it look like an HESC. Scientists have been researching these two types of formations because they are possibly effective. iPSCs are formed by taking a DNA-containing virus and injecting it into an HASC, which makes it have the same structure and characteristics as an HESC. But this may result in genetic changes and cause cancer through rapid division. But it doesn't harm the embryo. The second type, SCNTs, are formed through DNA being taken out of an HESC and put into a nucleus of an HASC, and then it starts subdividing. However, it causes controversy with ethics. iPSCs and SCNTs could be effective due to their controversies. Lara is going to be now talking about the ethical lens.
Stem cell therapies have increased at a rapid rate over the past few years, causing governments to have trouble tracking and regulating to prove their safety. This is risking the health of many patients. They're also skilled at seeming credible, making it hard for patients to confirm their reliability. The FDA found that 13 people in California were hospitalized after taking an unapproved stem cell therapy that failed to scan for diseases such as HIV. It is argued whether it is up to the patient or the business to confirm their reliability. However, researcher Leigh Turner suggests that the vulnerability of the targeted audience should put pressure on the research site, and that it is unethical to blame an unknowing patient for believing a truthful seeming ad. The use of certain unapproved stem cells can lead to other diseases, intensify the condition, cause injuries, or in the worst case, even death. There are three main types of stem cells when looking through the ethical lens. Adult stem cells. Although they have no ethical issue in usage, their issue lies in the fact that they are fully developed, meaning that they're not able to adapt as easily. The Mayo Clinic also found that they may contain abnormalities because of the environment, toxins, or errors from acquiring them. Induced pluripotent stem cells have the most prospective outcome as of right now. However, the tendency of uncontrolled cell division can lead to tumor genosity or teratoma. Lastly, human embryonic stem cells. Although there is no concern in their health usage, their concern lies in their usage. They're considered the most safe and effective due to the fact that they're pluripotent. They're derived from the human embryo in the blastocyst stage. There are many different viewpoints on them. They're seen as unethical due to the fact that it's taking and destroying the blastocyst, which is killing the fetus and therefore seen as killing a person. However, the Harvard Stem Cell Institute counter-argues this by claiming that the embryo is not yet fully developed. It is only 180 to 200 cells, meaning that it is not yet a fetus. They also say that clinics treat infertility through taking an embryo and fertilizing it with sperm. However, not all fertilized eggs are reimplanted into the person, therefore leaving the clinics to discard with the rest. This upset researchers, claiming that they should be able to use these soon-to-be discarded embryos. Now I'll be passing to Abby, who will be talking about the future
Before the use of stem cells may expand, HASCs and HESCs
need to have more research done on them so we can better understand how transplanted cells behave in the human body for them to be successful. Theoretically, this issue could be resolved by cloning an embryo using SCNTs, although this process seems to have difficulties leading into ethics. Due to the fact that embryonic stem cell usage is frowned upon since it harms the blastocyst, the best ethical solution is using iPSCs. According to the University of Nebraska, more research needs to be done on iPSCs and their constant regeneration. They show the most promise for solving ethical concerns. In addition to not causing ethical concerns like HESCs, using iPSCs would be safe, solving our health problem. Therefore, clinics should not have to create misinformation on their treatment. Although an effective option, looking through the ethical lens, is using HASCs, ethics does not support for this. Therefore, the next best solution, combining both lenses, is iPSCs. Even though scientists are trying to overcome this issue by blocking certain cell signaling pathways, we believe that with more information on iPSCs and their uncontrolled cell division, stem cell therapies will become effective in the future.
Grace, can you describe to us how the content of your team presentation was changed as a result of your group's discussion?
So we initially said that stem cell therapies were effective, for example, bone marrow transplant, and as we researched individually, we found more and more issues and more concerns. Like the ethical use taught us about the embryonic stem cells and how the ethical use kind of made us think differently about them, and the economical issue of not having enough money to fund this research, and the scientific one showed us that there was not enough info or research on these stem cells, so we changed our answer to not effective, and in the future will become so.
Okay, thank you. Lara, reflecting on your colleagues' work, which one had the greatest impact on your understanding of the problem your group identified?
I believe that the ethical lens had the greatest impact on our overall solution. The scientific lens contributed to the knowledge on adult stem cells causing health concerns. Therefore, our main solution would have been HESCs. However, due to the ethical concerns on usage of these and using the embryos, that it results in the destroying and killing of the blastocyst, therefore seen as destroying and killing of human life. Therefore, for this reason, we decided that the best solution would be iPSCs once we solved the issue of uncontrolled cell division, since it would be the most effective and therefore would have no concerns on ethical usage.
Okay, thank you, Lara. Anne-Marie, in what way did you improve your ability to work with a group as a result of this project?
My ability was improved because I was able to get feedback from everyone else on our solution. For example, Lara was able to help me with comparing my ideas of the solution, and we were able to come up together with our final solution that also led into Abby's, which left us no room for errors.
Okay, thank you. Abby, having finished your project, what, if anything, do you consider to be a gap in your team's research that, if you addressed it, would make you feel more confident about your conclusion?
Something we consider to be a gap in our research would be the fact that we do not have a set solution to the uncontrolled cell division. This would allow a more established and committed answer to the question. I researched a bit and found that they are trying to overcome the fast division by blocking certain cell signaling pathways that can cause cells to multiply rapidly. However, we believe having a clearer answer on how it would become possible would make our conclusion stronger.
Thank you. Another round of applause.